Okay, welcome everybody. We were so close to wrapping up chapter 11, just a handful of slides left to think a little bit about some genetics, and then we will transition into chapter 12 and thinking about how we can apply some of this for our own gains, I would say. There are all sorts of potentials when we think about having some understanding of genetics and then being able to manipulate that. And that's really, as we think with some of the biotechnology, how this chapter kind of sets the stage for our potentials to deal with all sorts of different genes from different organisms into bacteria or others. All right. So I believe I left off right here with the transposons. We sometimes call these guys jumping genes. When we think about eukaryotic cells in the process of meiosis, there can be some crossing over, and it's exchange of material from homologous chromosomes, so same chromosome number. It's not the same as what we're looking at right here, but it does contribute some variation when we think about crossing over, where there's the potential for one chromatid to have something that's slightly different than the parental type. Transposition happens either on the same chromosome, taking a gene and moving it someplace else, or we could think about potentials if we're looking at eukaryotes for transposition to happen from completely different chromosomes and those genes ultimately moving. And it doesn't matter who we're talking about. Typically, when transposition occurs, it's bad news. All right. There are some opportunities where it may not cause the disruption of a specific gene, and we might see some function. But thinking our bacteria, which is where we're at in the first place, the bulk of their genetic code is information in the first place. We would expect genes that are coding for specific functions to be grouped close together on those chromosomes, and in the process of transposition, we would disrupt that and ultimately alter the sequence, making it so that we've lost some function, typically is what we would think about. So the enzyme transposase causes DNA to get pulled together, and that's, as we see here, is where this disruption is occurring, as the transposon sandwiches itself in between this gene, causing it to lose that function. So I mentioned this briefly just 1 second ago. When we think about specifics, talking our prokaryotes, when we think about specifics that they may be utilizing, maybe enzymes for the breakdown of different sugars or something like that, grouping the genes side by side pretty much makes it so that we can produce these in a kind of a steady stream, if you will. So we've got our sequence of DNA up above, and we're looking at the messenger RNA that we're generating, and then the protein products, DNA to RNA to protein. We've talked about this before, our central dogma, the flow of information, all right? And we can see in green the structural genes A through D. All of this gets transcribed in our messenger RNA as we go through here and start the process of transcription. And in turn, multiple proteins, products can be generated, right? So as we look at this operon is this group of genes and the initiation, if you will, the promoter and operator that allows for the RNA polymerase to link up and to start reading. all right? So all of this together, and then a little bit upstream from that, some regulatory genes, things that we may think of as either being able to be turned on or turned off when we think about this regulation, so that we could keep the information flowing, or we could halt that, dependent upon really what's happening in the cell, all right? So a lot of different possibilities with this as we think about the ability and thinking, you know, a lot of these things kind of assembly line or sort of chain reaction situation. If we are able to make all of these simultaneously, we've got all of the business needed to break down these sugars or whatever it might be, all right? We've mentioned lactose with our friend E. coli, and if it's not present, we will repress, we'll shut off this function. There's no point in going through these mechanisms, generating the enzymes needed, keep it turned off. If lactose happens to be present, that's a different story, and we will allow these to be produced. It also depends on your circumstances. If you're in a environment, if you're in a flask that's got all sorts of different nutrients, you would be using what would be the simplest first, and then transitioning to something that might take a little more energy to deal with. So if glucose is present, we're not going to waste our time trying to utilize the lactose. We're going to burn through the glucose first. Once that's happened, then we shift over and start thinking, we need to go through this process and start making enzymes needed for that. So definitely some potentials with that, and we'll see lactose, I think maybe the next slide or two slides down the road, as we think a little bit about these ideas, all right? So here we can see another example with tryptophan, an amino acid, all right? Maybe you remember this amino acid. It's the culprit that they blame turkey having that makes you feel sleepy, right? Turkey does have some tryptophan, all right? But it's really not the turkey's fault that makes you so sleepy. If we think Thanksgiving, it's all the other stuff you've consumed. You got a super full belly, and that's what makes you want to sleep, right? My gut says you could go, you know, go to some sandwich shop and order yourself a turkey sandwich, and you're probably going to get more turkey on that sandwich than you eat at Thanksgiving. And you don't fall asleep after eating a turkey sandwich, right? Probably you don't, all right? It's not the turkey's fault, all right? So sorry about that, but I gotta say it. In the absence of tryptophan, the TRP, tryptophan repressor, disassociates from the operator, and in turn, RNA synthesis proceeds as we move through here. When it is present, we see this binding and tryptophan in turn binding to this repressor, and we stop that synthesis. So in this case, we are looking at the production of this amino acid. E. coli, our good friend E. coli, making tryptophan. As we look at all of the genes needed, build all of this stuff together, all sandwiched right together, right past this operator, right? So in this, we're not showing it, but this operon, right, as we're looking at all of this business, right? So there's 5 structural genes that are needed for the production of this, and they're all coupled together. And as we go through and produce it, this, as we see below, is a good example of some negative feedback. Once we have gone through this process and we start producing the tryptophan that the cell is needing, we're going to start accumulating that, and we get to the point that technically we're filled up, right? We've got ample amounts of tryptophan, and the tryptophan itself goes back and does the feedback inhibition, binding to the repressor that basically links up to the operator, and it's the stop sign saying, we're done, we're filled up, we've got ample tryptophan, quit making it. Once the levels get depleted, the tryptophan falls off of the repressor, and then we're back up to the top bit of this figure where the repressor detaches and polymerase is able to go through there, and we start the building process all over again. So it just depends really on the business going on inside of the cell. Are there ample quantities or not? And with feedback mechanisms, we ensure that we don't just constantly keep pumping this stuff out. Typically, that's what happens when we think about these sorts of mechanisms with some feedback sorts of inhibition. So here's the lactose that I was talking about as well. All right. So in this case, our repressor has bound and it's linked up if there isn't any lactose. Why waste our time and energy producing enzymes needed to break down sugars that we're not even in the presence of? If we don't have that food stuff, why waste our time, right? So the repressor's linked up when lactose is absent. And in this case, the lactose serves as the initiator, not linking up to stop stuff. Now it causes that repressor to fall off, and we go through and build the enzymes needed to deal with lactose. So the same but opposite of the previous slide when we look at what's happening. This building stuff until we get ample amounts and we repress, and in the opposite, we're repressed until we're in the presence of those sugars, in this case, that causes the process to start being transcribed to build those enzymes. So this is, I was hinting at this just a second ago. If you're growing in an environment where you've got multiple nutrients, all right, we would see these situations occurring where we're not going to be utilizing something that takes a little more work until we've gone through the easier stuff, all right? So E. coli feeding on glucose, because it's the simplest stuff to deal with in the first place. Once we start metabolizing the lactose, we're going to break it down into simple sugars, glucose and galactose respectively, and then pull those in to the metabolic pathways, all right? But initially, we're going to burn through the glucose. And then as we start to see those levels depleting, we get a little bit of time here because we need to go through the process of transcription. And remember, with our prokaryotes, transcription and translation happen almost simultaneously, right? It's all happening in the cytoplasm. We don't need to process anything. As soon as we start making the message, we can start building the proteins. But it takes a little bit of time for all of that stuff to get completed. Couple hours, as we see here, as we transition, building the machinery needed to deal with the lactose as we kind of maxed out the glucose that's available. Soon, potentially this week, if not, it's got to be coming right up. You're going to do some experiments in the lab where you will look at the potentials for bacteria to use some sugars, right? And these ferments, these phenol red broths, have one type of sugar in there, right? You'll get a handful of different broths, different sugars, but each of those tubes only has one type of sugar. And the idea is, can they use it or not? And how do they use it as you go through the course of time, all right? When we look at other stuff like nutrient broth, what most of your bacteria show up into your lab in, those things have all sorts of different foodstuffs in there, right? And they grow really well. And then we shift it over and we're like, okay, now can you grow just with this one type of food, right? That's really what we're truly trying to look at with some of the metabolism. What do you need, right? Or how many different things could you use? And that may clue us in, all right? Some, like our friend E. coli, can break down lots of sugars, and others not so much, all right? And that clues us into some details. You'll also potentially wonder, what does my unknown do? And it'll get incorporated into this mix as well, all right? With that said, if your unknown is fermenting something similar to E. coli, that doesn't necessarily mean that your unknown is E. coli, because there are other things that will ferment stuff similarly, but it doesn't mean it's not, all right? Truly kind of depends on those circumstances, all right? As far as what's up, all right? So this mechanism is really what stimulates this image right here, the transition from utilizing glucose to lactose. There's got to be some sort of hints or signatures that say, wait a minute, we got to do something different, right? We don't want to starve to death, right? So in order for that to make that shift from glucose to lactose, it's the cell's ability to sense its environment, to sense what's really happening metabolically, what's happening with my energy, right? It's like, looking at the power meter outside of your house spinning around, right? What's happening, right? The power goes out and maybe you go look and see, right? Did I blow a fuse? Did something happen, right? You want to know what's going on with the energy, right? So ATP is the energy that we think about inside of the cells, right? And as we start burning through this stuff, we could see the level of ATP that we're able to produce start to decline. As those fuel sources, as the glucose levels start to get depleted, we're not able to keep churning out maximum quantities of ATP. It starts to go down a little bit, all right? And as that's happening, this is what basically stimulates the transition. Some of the ATP gets manipulated. And typically when we think about ATPs, we pop off a phosphate when we're going through building processes. A single phosphate gets broken off, and then it almost immediately gets recharged as we're going through this process. But with adenyl cyclase, it does a double dose. It pops two phosphates off. Two of them together we call a pyrophosphate, and that causes this cyclic AMP, right? Adenyl cyclase causing this camp over here, or cyclic AMP, right? A single phosphate, right? And as it's linked together on here, this is that switch, right? And it in turn is what says we should start going through this process to transition over to the production of lactase. That's what we need to do, all right? So just depends on what you've got in your broth, right? What you've got in your environment, right? So in the absence of cyclic AMP, right, CAP doesn't do anything, doesn't bind, and we see transcription happening, right, at a low rate, right? When it binds, this stimulates the polymerase, and it basically says, we need to kick this into gear. We need these enzymes, right? And we see this happening arrow-wise, little bit and a big amount, right? So we shift it over to producing bulk quantities of it, all right? So this stimulating the activity, but we can also see, dependent upon circumstances, right? If the repressor has bound, it doesn't matter that we've produced this cyclic AMP. If there isn't any lactose in my environment in the first place, it doesn't matter that the cell is begging for me to transition. There's no sugar for me to use to begin with, right? No lactose. The glucose was there, but I've been using it up, right? So I start sending out these signals with the cyclic AMP saying, hey, we're going to start starving here, but there's no lactose, so it's unable to actually allow us to progress, right? That's the idea. And up above, if we do, up above, if we do have a little bit, we could see this slowly going and then kicking into gear as the glucose depletes. And then we've really kicked those genes in to say, let's build these enzymes, right? And like I said, if there's no lactose, why waste your time, right? You don't want to build those enzymes unless you truly needed them, right? So similar idea as we see this with these feedback. We're not going to go through all of these details with it, but the ability to turn it on or turn it off if we've got ample amounts, all right? So we could see some of these ribose switches that in turn allow things to either allow for the transcription to go, as we see up here, or shutting it off, right? And we can also think about translation. Once this has been made, we could start it or stop it dependent upon circumstances. So either the production of message or the reading of that message can be governed by these respective ribose switches that allow things to be turned on or turned off, right? So a little bit different, but same sort of an idea. In eukaryotes, there are some potentials where we can turn things on. Our enhancer sequence promotes transcription, and in turn, basically saying things are present. Let's go through with this, right? These activators that stimulate it, or we could see the opposite with repressors not allowing this to be transcribed. Not exactly the same idea, but pretty close, right? Okay, that's the end of that as I hit exercise. Shifting, find the right slide here. Shifting over to 12, we want to think about some details like how can we utilize this stuff? And I think this is truly very fascinating to think about all sorts of potentials that we really have at our fingertips to be able to manipulate genetic sequence. So microbial genetics, really thinking about these microbes, bacteria, or maybe some yeast cells and others. But we can take this and extrapolate that to thinking about eukaryotic genetics, our own genetics, as we go through and think about some of these steps in this process. So there's a lot of different things that we want to think about as we progress through here, but really the idea is looking at specific sequences of DNA, segments, genes, and in this first bit, this thermocycler is a machine that allows the opportunity for us to take that segment of DNA and, like a photocopier, it allows us to make massive amounts of copies of that sequence, all right? The thermocycler is where polymerase chain reactions occur, right? It's a machine that allows for, it's kind of like a scientific little easy bake oven, all right? It's going to allow things to get cooked up in there, all right? But this machine has a manifold that is, the little tubes go into that. It allows them to get heated up very rapidly, cooled very rapidly, and the process of PCR reactions can happen in a matter of a minute or so. You could go through the round of unzipping, replicating, having a new strand. And in the course of a handful of hours, you can go from one copy to billions of copies in that tube, all right? Massive amounts. You can't really do much of anything with a single copy of that gene. But if you've got a billion copies, now we could do some science, right? Now we've got the potential to actually do something with it, all right? There are lots of potentials, some of the biology labs, we try to show some DNA and we extract some by swabbing our cheek. And you can get the DNA from that little bit of scraping or swabbing of the cheek. You can get the DNA to precipitate out a solution and have a little glob of DNA that you can visualize. Thermocyclers, it's kind of like that, where you start off with a tube that's basically colorless liquid, and when it's done, you've got a little glob of DNA, a cloudy sort of precipitate blob of replicated sequence that you can actually see, all right? And then, like I said, we've got potentials to do all sorts of things with, all right? So the PCR reactions were ultimately made possible with the discovery of some organisms in Yellowstone. I think we showed some of these images early on with some of the hot pools. Some of these microbes that can thrive at super warm temperatures have enzymes that survive and are still functional at super warm temperatures, all right? Not survive, but are active, all right? And in turn, that opened up the avenue for being able to manipulate this stuff in the first place, all right? So we could get all sorts of details. We can also utilize PCR technology to give us some information that might help for types of organisms that are more difficult to cultivate in the lab. The example up here is some chlamydia. Chlamydia trachomatis is a sexually transmitted infection. It is something that is kind of top of the list, at least in the US, when we think about sexually transmitted diseases. And chlamydia is an organism that we call an obligate intracellular parasite. These organisms have to live inside of some of our cells. You can't just put them onto a nutrient agar plate and expect that chlamydia is going to grow, because it won't. It's got to be in a living cell, all right? That requires completely different culturing techniques. You have to have a living cell line, human cells, in order for it to be cultivated. Definitely takes more work, all right? And we can see a couple of these cells that are infected with the chlamydia and a bunch of other normal human cells in this culture, all right? So taking a sample and using PCR can kind of cut out that middleman, if you will. There's no need for us to do any cultivation. We can do some PCR techniques and actually amplify the genetic material without the need to culture, all right? There are numerous ways PCR reactions function. Lots of times we do think about growing something up, extracting the DNA, and then using PCR. But there are other techniques where you can take a sample and run through these reactions and get more of an overview of what's out there, all right? Potentially thinking maybe an opportunity to detect biological warfare agents without the need of cultivating all sorts of things to see. We could take some samples and run it through this and amplify what's there. And if those culprits truly are there, their signatures will show up, all right? Which is impressive, all right? There have been numerous advances with these, and it depends truly on what you're going for as far as how that may function, all right? So for genetic engineering, for biotechnology, there's all sorts of things that we think about the opportunity to manipulate. But when it comes to humans, the first stuff that we think about with genetic engineering was insulin. It's something that diabetics, you need this stuff, type 1 diabetics, right? And in turn, before we think about genetically manipulating this, we would harvest it from the pancreas of an animal, pigs, typically, all right? And pulling that out of there, and that's where you would get the insulin you needed. With biotechnology, the thought was, why do that? Can't we just take the gene and incorporate it into a bacterium and grow it up? There are a handful of things we need to do, and we've talked a little bit about it before. We'll see more of it down the line in a minute, but you can't just take the DNA from a human cell and pop it into the bacterium like we're showing right here, right? It takes a little bit more, right? We need to manipulate it. And we talked about eukaryotic versus prokaryotic DNA. And in eukaryotes, there's extra stuff, those introns, the spacers in between the genes and flanking the genes. And those things in the process of transcription get copied. And then before we get to translation, the DNA gets or the RNA, gets modified, and then we build the proper stuff. All right. So you can't just pop the DNA out and expect that it functions. We need to manipulate it first by taking that message and using reverse transcriptase to make a copy of complementary DNA. Almost always, when we do some of these manipulations, we utilize plasmids, not 100% of the time, but pretty close, all right? And plasmids are some extra chromosomal material. They're easier to pop out and to manipulate and to pop back into a bacterial cell. Trying to do that with the bacteria's chromosome would be a problem, right? But the plasmid, the cell can live without it. And typically when we think about plasmids, it might live a little better if it's got it. Now it could resist an antibiotic or something else, right? In this instance, it might do that, but it also pumps out insulin, all right? That's the idea. So if this plasmid's able to get into the bacterium, as that bacterium grows and as it replicates, in the process of replication, it also, those plasmids receive cellular clues. It's like, oh, sweet, we're going to replicate. We'll replicate our plasmids too, so that in these two new daughter cells, we're both going to have plasmids, right? And in the process of just reading this material, whether it be through the process of replication or just looking at the specific sequence, we read these genes and produce the respective product. In this case, these cells, as they're cultivated, start producing copious amounts of insulin that's released out into the media. And we could collect that, purify it, and sell it, all right? That's the idea. And all of this, once we've popped something from some cell into this plasmid and got it into a bacterium, we say this is transgenic. It's got different genes than the bacteria itself, right? And there are all sorts of transgenic things and genetically modified things, which are transgenic if we manipulated things, right? In the United States, if you eat food, you're eating genetically modified food, all right? Unless you are making a serious effort to avoid that, and I think good luck with that, all right? You are consuming genetically modified stuff, all right? All sorts of agricultural products have genetic manipulations within them, all right? And lots of times we think about these crops having higher yield, higher, different nutrient contents as a potential that we think of as benefits, all right? There are maybe some potential issues with some of that, depending upon what's been manipulated, right? They don't necessarily tell what sorts of things they have done, right? And in turn, maybe there's a gene that's been pulled out of some organism that you happen to have some allergies to, right? And now it's been popped into something that you're not allergic to. And then you consume that product and you have an allergic reaction. but you're like, but this tofu shouldn't have made me sick at all, right? But maybe it has some other protein in there, right? I don't know this for sure, but I feel like sometimes they cover, different companies cover their bases when they say on there, this product was manufactured in a facility that also deals with tree nuts or dairy or whatever it might be. Some of it is, maybe they are working with other things, or is other parts of it that this product has some tree nut protein incorporated into it, and we cover our base because this facility processes that stuff too. Or deep down, it's just in your stuff, right? I don't know if that's accurate or not, but it seems like it might be, right? Now the warning's there, right? If you've got allergies to specific things, you probably look at labels, right? You probably want to know, right? What's in this, right? Or not. So the DNA polymerase that came from those hot pools, we call it Taq polymerase for Thermus aquaticus, and it's high heat stable and can go through this process of making these copies. But we need to get the specific stuff out in the first place. And so bacteria, I've mentioned repeatedly, they're pretty stinking good recyclers. They like all sorts of things from the environment, and then we'll pull them in, maybe use them. Maybe I could eat it. Maybe why build all these nucleotides when I can just pull in some of this stuff, right? Transformation, the uptake of naked DNA, right? Well, maybe I pull some of that in, and now I want to cut it into pieces, right? Restriction enzymes are like molecular scissors. They're little bits that chew up DNA, right? But they don't just randomly make cuts. They're specific, all right? And they make cuts by looking at specific sequence of DNA. They see whatever assortment of these nucleotides, and this enzyme sees it and says, oh, I will cut that right here, all right? A different enzyme sees a different sequence and makes a different cut. And you could potentially take a segment of DNA and expose it to a handful of multiple segments of DNA, but expose them to different enzymes and get them cut up into different pieces, and then see what those cuts really look like. All right, that's some DNA fingerprinting that we'll look at in a second. All right, so the way that it makes these cuts is different, right? Based off of the nucleotides. And there are boatloads of enzymes that are these scissors, all right? And more of them being discovered, all right? Some of them make cuts that are jagged. We call these sticky ends, where there's some overlap. And when it comes to some molecular engineering, usually the sticky ends are easier to work with, and you can get things to go into those plasmids properly. You cut the DNA of interest with these enzymes, and you cut your plasmid with the same enzyme. And now the sticky ends match, and my DNA of interest can go right into that plasmid, all right? There are other scissors that make straight cuts. We call those blunt ends. And that you can do when you're trying to manipulate stuff, but the potential for it to go flip-flopped around is there, all right? And now you've got something not in the proper orientation. That sets the stage for problems, all right? There are ways to make it work, but it is definitely a little bit more labor intensive to make these guys work versus the sticky ends, all right? Where they can match back up, all right? A lot of different things, all right? And maybe we're looking at, we've extracted DNA from some organism. We could take that DNA and we could do this digest, a restriction enzyme digest. We've got one sample of our DNA with one enzyme, and we allow it to go through there. And likely there's going to be some sequence that will be recognized and cut. But the question is, how many cuts? How many specific sequences are in that DNA that these enzymes can cut? Maybe we get half a dozen. Maybe we get more than that. Maybe we get none, right? But we get some information, right? And if we employ a handful of those enzymes, that gives us a pattern that we could look at, and then we could compare it to a different organism and do the same sort of thing and see what sorts of cuts are truly happening, right? And giving us, as we run it out on a gel, the ability to see these bands and get a fingerprint, if you will. So here we've got a plasmid, and this is an artificially constructed plasmid vector, PUC19, and it's one that's utilized for cloning, for putting DNA in there, all right? And there are right up in here, all right, a handful of sites on this sequence of plasmid that different enzymes can come in and make their cuts, right? So we've got a whole suite of genes that we may cut out with an enzyme, and this plasmid, dependent upon the enzyme I used to cut my gene out, I use the same one on my plasmid. Then the sticky ends match, and now this one that I cut with Echo R1 fits right in there. Or maybe I used the HIND3, right? And I cut the plasmid with the same thing. It's right back to this. Whichever one I used to cut my gene out, I use the same one to cut my plasmid, and it just happens that I've got a whole bunch of them right here, so my gene's gonna fit right in them, right? Right where I made these cuts. And this would be the insertion site where it can go in, right? Right up above it, we can see a LAC-Z, right? These guys, there's a handful of things in here, but lactose it can utilize. We've got the genes for it. Ampicillin over here is another gene, different part of the plasmid. Ampicillin resistance is a way that we can grow these guys up and truly see if they actually worked or not. If you've got a petri dish full of this antibiotic, bacteria that aren't resistant to it are going to die. And whatever we're putting this plasmid into would be sensitive to that antibiotic. But if the plasmid got into the bacterium and everything's functioning properly, this gene will get turned on. Our bacteria can now grow on the plate. And if the insert happened properly, the LAC gene gets turned on, and now these colonies show up typically as a color, blue color, that shows not only are we growing on the plate, I got the plasmid in me, I turned blue, and my insert must have went in there properly because my LAC gene is functioning like it should, and there you go. I could pick that little blue colony off, culture it, grow up mass quantities of whatever it was that I was trying to get in the first place, all right, in this respective plasmid, all right? And there are all sorts of things that we may like to try to get with something like that, all right? So this is molecular cloning. This isn't making a little copy of myself, all right? That's not the type of cloning we're talking about, all right? And when it comes to cloning, microbiologists have been doing cloning for a super long time, all right? Not making ourselves, all right? This type of molecular cloning is taking a gene and putting it into something else. The same picture I just showed you a second ago, all right? But instead of just this one bit, we could take the entire genome from something, and we could chop it up with those scissors, and we could insert each of those pieces into a different plasmid, and we could grow them up. And that's what we're looking at with some of this, right? In these instances, we're putting it in, inserting it. We've got our gene for resistance, and we've got colonies that show this, right? In this case, white colonies show up with the recombinant plasmids, and the blue colonies show up as growing, but we didn't get the gene in as we wanted. So it was the opposite of what I was just saying with the blue ones. But either way, you get a color difference, right? In one of the microbiology labs, a pathogenic micro lab that I'm teaching right now, we grew up a handful of different E. coli strains that had some plasmids incorporated into them. And they were all kind of a rainbow of colors, some blues, some pinks, some greens, some orange, each one a different plasmid giving a different pigment as it grows up. And it was kanamycin as the antibiotic instead of ampicillin, but similar sort of an idea, right? And my students did a little streaking for isolation just to see that as it went through there. And this was kind of a pilot study to see, is this something that maybe we could think about in intro micro, getting some nice colors when you do some of the streaking, I think makes it look pretty cool. And it's also pretty simple to look at those plates real quickly and see bright red colonies or purple colonies versus some that are not so impressive and just kind of cream colored, all right? But that's sort of like this right here. All right, I'm going to stop with that. We will pick up with some ideas about making a library of different parts of the genome on Wednesday. Farewell.